to see an opening slide I usually use for my talks in Singapore. Somehow I neglected to swap out the background. Yeah, but it's a good reminder. It's a good reminder. I'm actually based in Singapore. That's where Google's Asia headquarters is. And I am a developer advocate in cloud. I think that might be on the next slide. Ah, yeah, there we go. Yeah, and so today I'm going to be talking about deep learning specifically for applications in life sciences. By background, I did my PhD work in human genetics department inside of a medical school, focused in bioinformatics and genomics applications, working with DNA microarrays, DNA sequencers, et cetera. These days I tend to be more focused a little bit broader on agricultural applications, and I'm going to talk about some of that. Yeah, and we have some cloud services offerings in that space specifically, but I'm going to keep this mostly very much a science and technical talk, and not so much talk about cloud services. All right, so I should also point out that a lot of the work I'm presenting here today is not done by me. I'm just a talking head. Most of it's coming out of Google Brain. This is one of our research units at Google from DeepMind. They have a health division. Verily and Calico are sibling companies inside of Alphabet's holdings. Alphabet is the parent company of Google. And I'm specifically in the cloud division of Google, and I focus on the healthcare type applications. So there's a lot of collaboration going on between these companies and different teams. We publish together and we make software together. All right, so just as an overview, what we're going to be talking about today, I know, how many of you have been in this track like all day and gotten the same introduction over and over again about machine learning? Okay, good. So I only give you 2 slides. I figured we can skip that. I knew I was at the end of the day. Thanks for sticking around, by the way. So we won't go too far into the intro. Why this is happening now, this might be a bit of a different bent on why deep learning is showing up right now. How we're applying it specifically for clinical diagnostics, and then some clinical genomics and agricultural genomics examples of applying these deep learning technologies. Okay, so a lot of what I'm going to be showing, actually everything I'm showing you today is based on a particular deep learning architecture called the convolutional neural network, specifically for vision examples. Essentially what these are doing are taking low-level features at the pixel level, depicted at the bottom of this slide as raw data, and through labeling of features, we can essentially pass these pixels through a series of tensors or layers in a neural network data structure to build aggregates of the raw input, and then aggregates of aggregates, or composites of composites, composites of composites of composites, and so on, all the way up. The higher the level of compositing you do, the more abstract a pattern can be represented. And so this enables the machine to represent arbitrarily complex patterns by having lots of layers of these tensor representations or dimension reductions of the original input data. That's in basically learning mode, and then you can go into an inference mode. If I had my GIF animation, which I don't because it's a PDF, you would see that basically these cat and dog images flow through a series of layers. And there are effectively some coefficients that are added during training that allow estimates of what the image contains to be made, and we can discriminate between cat and dog photos. What's really happening slightly under the hood is that it's really probabilities that are being extracted, and we would just take the maximum probability and label a photo. In this case, I have no idea where this label came from. I just love this slide. I found it in another person's deck. Of being a tiger dog. I guess it's a Halloween costume or something. It's great though. Essentially what's going on though is, as I was talking about, there are multiple layers that are being used to label this photo. This is a famous architecture from Google called the inception model. And you can really just see there are several essentially very similar layers added in series, each one of which is doing a further dimension reduction on the data, and then leading to that final classifier step that you're seeing on the far right there that gives the probabilities of a set of labels. Something to note here is there's actually some side paths off of this network, and so those can be used for using the network for alternate applications. For example, you can also remove off this final layer to do something called transfer learning, to retrain the inception network to recognize arbitrary other classes if you give specific examples. And if you guys were here for Paige's talk in the last session, she was talking about training on 5 examples of C3PO and so on. That's essentially what this is for. It's for transfer learning. Let's skip that. We're using the same kind of technology across all of Google's product lines. There's an application library called TensorFlow, developed by Google. It's now open source. It's the most popular deep learning library on GitHub and pretty much by all the measures. And it's also the most, one of the most popular libraries that we're using internally at Google. So TensorFlow is being used everywhere. Deep learning is impacting all of our products, from search to Android to ads and VR. All right. So why is this deep learning technique becoming popular right now? Essentially because, for historical reasons, it used to be that the accuracy of classical statistical models, so models with fewer parameters, but they were making estimates of the values of those parameters in a sparse data environment. So way back in the day, we didn't have very much data, right? And because of Moore's law and the increase in areal density of storage and transistors through photolithography, we have more data storage capability and just more data as a result. But in the past, that wasn't really so true. And so in the context of less data, we had to do some hacks of assuming, like, we know natural data approximately follows a normal distribution in some cases, or a Poisson distribution, or hypergeometric, or some number of other distributions that are known to describe natural phenomena. And if you don't have very much data, it's actually good to use these approximations to get an understanding of what the shape of data are to build a classifier, let's say. And that's what I'm talking about here when I give this y-axis of accuracy, okay? And neural networks, which have a lot of parameters, require a lot of data to perform well, because they're trying to do a very high precision modeling of data to make an accurate prediction. So back in the day, we didn't have much data, and these fewer parameter models did better. But as we had added more compute power and more storage through Moore's law, which basically describes it as doubling every 18 months or so, we get to a situation where there's actually a tipping point, and we're on the other side of that, or crossing point, and we're on the other side of the crossing point now, where we have enough data that actually these neural networks that require a lot of data to perform well have achieved liftoff. And so now that they're now outperforming the classical models because we're able to effectively run them and we have enough data to train them. This is another example or this is an example of how these networks are performing. So there's a competition that runs every year called ImageNet, where there is essentially a large number of images that need to be binned into several classes. Humans are performing at about a 5% error rate, and for a long time, machines were not really doing any better than humans until these deep learning techniques showed up, where we could have lots of layers of tensors to represent arbitrarily complex patterns, right? And then the computers started to perform well around the year 2011, when it was recognized that GPUs and the vector processing that they were doing for motion picture and for entertainment industry, for games as well, could be repurposed to do vector processing, which is the specific sub-operation of matrix or tensor processing, which is what's being used to drive these deep learning networks. And so by having more of this vector processing ability, we could build deeper networks, which led to more ability to represent these concepts, higher order concepts, which led to better performance. All right. Okay, so those convolutional networks aren't just good for recognizing pictures of cats and dogs or Star Wars robots, but also for doing useful industrial work, in this case coming out of the clinic. So I'm gonna talk to you about a use case that's happening right now. There's a global pandemic. It's diabetes. Approximately 5% of the population in the US is diabetic, and it's approaching this globally as well, as diets are westernizing. One of the side effects of diabetes is diabetic retinopathy. So retinopathy is a pathology of the retina, essentially what's happening is the pathology of the blood, where there's too much sugar, too much metabolites in the blood, are causing some small, basically scarring on the retina. And as through blood vessels bursting, the capillaries bursting, it's a very delicate tissue. And when that happens, the blood starts to rot, and then there's a scar that gets created on the retina. And then that scar, where the photoreceptors were before, these cells that receive the light and send it to the brain, they're no longer functional because the scar is there. And so that's where the blindness is coming from. What's done today is that there is some well-trained humans that go through medical school. They're ophthalmologists. They need to look at some images of retinas to do screening, and if you can detect that there's this retinopathy beginning to occur, you can intervene and prevent the blindness. But in the case that the damage takes place, you can't actually reverse it, and so once the damage is done, it's done. And you didn't prevent it, then the damage is done. So right now, there's a problem because these humans take a long time to train, right? They go through 6 years of medical school plus a residency. So it's a very not scalable solution, and they're also pretty expensive, and they only have so many hours per day they can look at images, and it takes some time because it's highly technical. And so this means that we're not able to effectively screen all the patients that we need to. So it sounds like a good application for image processing if we can find a way to do good classification on the images. Here's a picture of what these looks like. It's probably kind of hard to see. It looks a bit washed out. But you can see on the left side, hopefully, that this is a healthy retina. And on the right side, there's some small hemorrhages. That's what I was talking about, where the blood vessels have burst, and that leads to the scarring tissue. Essentially, the classifiers, whether it's a human or a machine, are looking at this image, and they classify into no diabetic retinopathy all the way through what's called proliferative, which is extreme retinopathy in the diseased state. And so the goal is to basically get a machine that can perform as well as a human or outperform a human to offload the work, right? Make it more scalable, make it cheaper. It turns out that if you look at a bunch of human graders, though, they're actually wildly inconsistent. So I've highlighted some of the more extreme examples here with black bars for the rows, where each of the columns is a human and each of the rows is a sample. So we asked a bunch of ophthalmologist graders to grade multiple samples, and you can see that sometimes there's some underestimating, where most ophthalmologists are saying that the image is proliferative. That would be the lower black row. But we've got one of them that's saying that it's mild. And then in the converse case, you've got other samples where there's an overestimation of the condition, where most of the ophthalmologists are saying that the tissue is healthy, and one of them is saying that it's actually very, very diseased. So they're not consistent between one another, and if you show the same image to the same ophthalmologist, they're not necessarily consistent with their own previous ratings either. So this is problematic. We can, however, take a bunch of data. If we have 54 ophthalmologists, this is a data set we created, and have them grade a whole bunch of images. This is paying ophthalmologists to basically label data for preparing a supervised training to build a model. And then we can do the standard training test, validation data sets to train a convolutional neural network to look at these images and output the correct score, from score of 0 to score of 4 in this case. Once that's done, we can look at the precision and recall of the model on correctly giving the right score and not being confused by the images. And you can combine these precision recalls or area under curve ROC type plot into an F score, which is a combination of sensitivity and specificity. And you can see that the algorithm can outperform humans. This was published in the Journal of the American Medical Association. And it's just one example of this type of medical imaging application happening across all of medicine. I'll give you another example. This one is a bit different. It's involving some tissue biopsies. And it gets, and I'm highlighting here that problem I was just describing about under and over diagnosis. So in this case, for breast cancers, there are multiple different types. You could think about these as grades of severity like we talked about before. And if you look at the pathologist classification of a given pathologist versus a consensus of pathologists, so we basically find images that are unanimously graded by a panel and then compare that unanimous grading to another pathologist to see do they agree with the established consensus. You can see that there's sometimes some underdiagnosis. So there's a consensus that says there's a cancer of some given level of severity, but it might by a single testing pathologist give it a different score that's actually more mild than you would have expected. So this could actually lead to progression of a disease that would not otherwise be necessary, right? And sometimes there's an overdiagnosis. So it could be that there's something that's not really very severe, but it's overgraded, and then extra therapy is taken, such as a mastectomy, where the breast tissue is removed when it didn't necessarily need to be. So sometimes this happens as well. And we don't want to fall into either of these cases, right? We want to stay on the diagonal and correctly diagnose everything. This is a slightly different model, where sometimes some information about the cancer, if we're going to look at a biopsy of it, so this is where you basically cut a chunk of tissue out of the cancer, you slice it into a very thin slice that light can pass through, it becomes transparent. You put it on a microscope, and then you can scan the microscope slide. You put it on the slide, you scan it with a microscope to get various levels of magnification features depending on what type of lenses you're using. And the reason we do these different magnification levels is because certain features of the cancer tissue is evident at different magnification levels. So for example, at a very high magnification, you might see that there are some aberrations inside the cell itself, inside the organelles. Maybe there's something strange going on inside the cell, and you can see that structurally. Other cases, you might be looking for something like more macro features, like blood vessels being developed where they shouldn't be. Because the cancer is trying to recruit blood so it can feed itself, so it can grow. And that wouldn't be obvious at a high magnification. So what we did is we took basically these images of the cancer at different magnification levels, and then predicted whether the center patch of some pixels relative to the surrounding pixels is cancerous or not, based on labeling from some physician. So there's like 23 million of these patches were extracted at all the mag levels. And classified. You can see the original image here on the far left. The middle slide is the human pathologist. They went ahead and drew a bounding polygon around the cancer cells, and they were given unlimited amount of time to do that, to correctly label what they thought was the cancerous regions versus the non-cancerous regions in the slide. Again, at the various mag levels. And then that was used to build a predictor based on an ensemble of these different neural networks of the different magnification levels to predict whether cancer was present at any given point on the, well, in this case, the most zoomed out picture of the biopsy. And you can see that the machine is accurately, well, it's getting the same answer that the humans gave it. So the model is correctly modeling the human predicting capability. And then again, based on this underdiagnosis, overdiagnosis, we can see that the F score is actually slightly better for the machine than it is for any given pathologist. So this is, again, a way that we can use these deep learning image processing systems to process more images in less time at lower cost, running 24 hours a day in the data center rather than having to train more physicians for 10 years apiece. All right. Great. Now we'll talk about some genomics applications. So this is really my specific area of expertise. And I want to start with a slide. I mentioned earlier that these deep learning networks are becoming effective because of Moore's law, which is creating more data and creating more computing capability. That's plotted here in a purple line, so you can see that the slope of Moore's law is plotted in purple. And I'm also plotting some other data variables. This is related to producing DNA sequence, so we can think about how many cents it costs per base to sequence, and then we can calculate what the cost is of a human genome, given that there are 3 billion DNA base pairs in a genome. And you can see, and this is on a logarithmic scale on the y-axis, and time is presented on x. And you can see that it's exponentially decreasing, but remarkably, it's actually the cost is decreasing, or the throughput is increasing for DNA sequencers at a rate that's outstripping Moore's law. So it's going even faster than the computers, because we're developing some really interesting nanoscale technology for reading DNA. I'll come back to this later. I'll get now into some basic genetics. I don't know how many of you guys remember this. You might have seen some of it in school. It's a really simplified model, and I had fun with the emojis. You can basically think about historically, before we had any genetic information, we might expect, like, if we crossed a male and a female to produce an offspring, in this case it's a girl, it's basically random whether she turns out to be I guess it's a flamenco dancer, maybe, or a coder. But in reality, there's something kind of more complicated going on here. It's not really just the crossing of the parents that produces an offspring that's like a combination of them. There's also a whole bunch of environmental factors. So the first piece is a prior, where there's some genetic basis, a template, effectively, that can then be modified by the environment. So for example, if we were talking about, like, skin pigmentation, right? There's going to be some base level of skin pigmentation, and then based on exposure to sun, you might increase the melanin content of the skin because it's responding to prevent UV damage, and the skin gets darker. Or you eat some food that makes your eye color lighter, or you get exposed to different languages and have different phenotypes related to that. So there's a whole bunch of environmental variables that go into producing the downstream phenotype that's observed for a given individual, given organism, on top of the genotype. Getting a bit higher resolution into this, there's actually some DNA coming from the parents. So I'm representing this as pairs of chromosomes that are coming from the mother and pairs from the father. So what happens is, and those came from their parents, right, recursively up back to the beginning of the species. And what happens is this child is getting 2 chromosomes also, typically, when it's not an aberration. One from the mother, one from the father. So effectively what's going on is we can choose all possible combinations of pairs from the mother and the father. That produces some grid of children that are being sampled from for any given child. Only one, unless you're dealing with identical twins or triplets or something like that. And then that goes down to the next level, where you apply the environment to get some downstream phenotype. Okay. So that was a simplified diagram I was showing you before, but in reality it's more complicated than this, because there's actually 3 billion positions in the human genome, times 2 chromosomes per parent. And for any given human, there's approximately 3 million of those positions that are unknown. So they differ between you and any one of you and me at about 3 million positions, and that's why we're not clones of one another, because of this genetic variation. So it's useful to know for any given human which of these positions are, according to this cartoon I'm showing you here, not blue or are a variant. And if they are a variant, you actually want to know what the state of them is, so that you can understand that genetic preloading leading into the environmental influence, right? So this is the hereditary component of phenotype. And as the cost is coming down, effectively what we're going to be doing, and you can already see this through some of the, well, I guess most strikingly in the consumer genetic testing market, like 23andMe or Ancestry.com or some of these other services that are available, where people are beginning to get some low-resolution information about their genetic data. And ultimately what will happen is these genomes are going to become cheap enough that they're just produced on all newborns. To give you some sense of scale here, this is an old slide, actually. YouTube is getting about 300 hours of HD video. This is pre-4K, so it should be a bigger number now, which is equivalent to about 6 genomes per minute. And the number of births in the US per minute is about 5. And globally, I think it's under 100. It's like 50 or something like that. So it gives you basically, you know, we're already there, internet scale-wise, to accommodate this level of data. It's not really an issue to collect it and store it on media in data centers, but analyzing it is a problem, which is why we're talking about this genome analysis problem today. Yeah, we handle like huge volumes of data. All right, and this is something else to note here. Coming back to this cost of genome sequencing figure I was showing you earlier, you know, we've all gone through, yeah, looking at the age of the people in the room, we've all basically seen the internet come online, right? And we've seen like dramatic changes in society. These days, you know, a lot of self-driving car stuff we can see coming. The rate at which those disruptions are happening, and we can see the world rewiring itself as more information becomes available and as we're connecting everything to the internet, that was happening at Moore's law rate, which is shown in the purple line again. The genome sequencing is actually we're producing data even faster than that, which means that the rate of disruption in medicine, as these data come online and we can understand what's going on in here by applying deep learning techniques or whatever comes next, is going to be even faster and more disruptive. Yeah, so get ready for that. Okay, so if we're going to try to pick an entry point area for applying this type of technology to life sciences problems, it's particularly suitable to apply it to genomics data first, because we have lots of data, like I was just showing you. Ideally, you want many samples for training to build a good model. You also want high quality data. And you know, labeling photos of cats and dogs on the internet, people aren't really so paranoid about making sure they got the label right. But when you start talking about life and death issues of what happens to a cancer patient, people get pretty sensitive about making sure the labels are correct, right? It's important for all sorts of reasons. Maybe slightly below like military data at the state level, but it's quite important. It's also so we need to know we have high quality labels, and that checks that box because it's medical data. We also want to make sure that we're working in an area that really needs deep learning and is a hard function to solve. So we know that there is some genetic predisposition for the phenotype, right? There's the genetic information plus the environmental information that lead to that downstream phenotype, the flamenco dancer and the coder I was showing you earlier. But we don't necessarily know what that function is or mechanically how it works, but we know there's something there because we know there's hereditary components to all phenotypes. And so we want to model that function. And we also want to try to attack an area where it's key that deep learning will be necessary to do it. So other traditional models that oversimplify, like I was talking about these classical models, are not really working so well. And so variant calling, which is labeled as SNP and indel, or insertion and deletion calling, from next generation sequencing data or DNA sequencing data, is a particularly good problem to work on because we've been trying to solve this problem of identifying for any given individual where they differ compared to another individual for several decades now, and the progress is pretty much plateaued, like we were seeing in that ImageNet example I was showing you earlier, where the performance wasn't as good as humans and then had a dramatic drop. we're trying to basically create that same thing for variant calling. To give you some sense of what that problem looks like, it's essentially trying to identify locations in the genome where we have a reference. So I'm showing this as this bottom section of a bunch of ATCGs. Top row is a reference. So we want to know where a given individual differs relative to that reference. And you can see that there's in the center here, there's a letter C highlighted in blue on the top row. And you can see that there's a G for the next three, what are labeled as reads, read 1, 2, and 3. And so we basically wanted to detect the individual as a G at a position, whatever the coordinates of that are in the array, right, the index, relative to the reference. So why do we have 3 reads? Why can't we just observe it once for the individual we're comparing to the reference to be done with it? There's an oversampling that's happening here. And the purpose for the oversampling is that we actually could make a single observation that's very high fidelity, and we know specifically there's a G there with only one observation. The problem is if we want to use this particular type of chemical assay to do that, it's very expensive and it's very low throughput. So it's actually much cheaper in terms of the number of base pairs you can sequence per dollar if you use some very highly parallel, multiplexed chemical process that is very error prone but produces tons of data, and then you feed all that tons of data into the machine to basically do some signal processing to identify the errors, wash them out, and figure out that there's something there. That's way cheaper, and that's where that faster than Moore's law curve I was showing you earlier is coming from, is from this parallelization of the DNA sequencing technology. Okay, so that's why we have to do that. Yeah, so that creates, it basically pushes the problem downstream. We've made it cheaper on the DNA sequencing level, but we've created a bunch of data. Now we need to computationally resolve it to figure out what we're really trying to measure, right, through this signal processing. Okay, yeah, and then we figure out what's the real sequence, and then we're done. There's a whole bunch of errors that get introduced by this parallelization process. It's super noisy. Some of it's being introduced by processing. So sometimes, or data collection, sometimes the sample is degraded, like it's not kept cold when it gets collected, and it oxidizes or it starts to rot. Sometimes there are artifacts that get introduced by the sequencer itself. So it might be biased to say there's a G when there's an A, or something like that. And that's a chemical artifact that shows up dependent on the specific generation of the technology that we're talking about, and that needs to be modeled. So there's been several decades of work done to try to build models that address these problems. If you look at the literature and survey it, you'll see that there's a whole bunch of classical statistic methods that are making some approximations and assumptions and fine-tuning by some human experts of models where those approximations are known to be inaccurate, and it takes unscalable, like human labor of PhDs who are trained for 10 plus years to be able to build these kind of things in order to get any kind of yield from the algorithms. Starts to sound kind of like the ophthalmologist I was talking about earlier, right? It's a very similar kind of situation where they're handcrafting. And so we want to find a way to build a general purpose hammer to kill the problem. All right, so here's what we've been working on. And this code's available. I'll show you some of that in a moment. Or I'm not going to show you code, but I'll give you a link to it. Basically, we take the data type. Okay, so we've got the reference genome shown on the, I'm going to walk you through on the left. We've got a bunch of reads that are of various states at a given position. And we're basically doing a sliding window, okay? From that, we build something called a pile-up image. So it takes all the reads that align at that position, where that's the best place they align. We're doing something like a substring string search to find the best position a read came from. And then that matrix of the reads, plus some other data, like what's our confidence that we believe there's an A here when the machine says it's an A. Are we sure it's an A? Do we think it might be an A? Are we not sure at all? All that information is captured. And so that builds a tensor here. So I'm showing you the layers of the tensor. And then that goes into a convolutional neural network. So basically, we're then tiling across the genome, looking at any given context, and making a call. So in the case of a human, because there's one chromosome from each of the parents, we can have really only 3 possible outcomes. We can have, hey, this is exactly the same as the reference. There's no new information here. It can be one of those chromosomes is not the same as the reference, and the other one's the same. Or you can have the situation where, hey, they're both different. And what are those different states? And so those are the 3 labels that we want to give as the output of this convolutional neural network. And yeah, we're basically interested in doing this to identify all the variants. All right. Here's a diagram of what that looks like, just doing some color encoding of the pileup image I was just talking about, where different colors and different levels of alpha are used for various attributes of the tensor, just to give you some idea of what's going on. So here in the center one, you can see that black kind of vertical bar. There's a deletion that took place there on the individual we're looking at relative to the reference. So there's a hole. The DNA is just missing. You can see on the top image, actually, I think we played with the alpha channel on this one, where you can see there's a strong mismatch highlighting. Not all the reads are mismatching. Look at that. Like, some of them are not, so it's probably a heterozygote where one of them matches and one of them doesn't. Yeah, just some visualization of the data input. Not surprisingly, as I was telling you, this is a convolutional neural network. It's using the same architecture I was showing you before that's being used for winning ImageNet and, you know, categorizing Star Wars droids and everything. It's the same type of architecture, where we're really just taking the final layer on the far right and removing its notion that it needs to classify objects into the ImageNet categories, and instead telling it, hey, I want you to classify into homozygous, alternate homozygous reference, or heterozygote, the categories that are of interest for this particular variant calling application. Okay? So we can leverage all the training on this neural network that was built for another application to reuse it for this new application. Let's skip that. This seems like a repeat slide. Okay, and yeah, it's pretty awesome. So there's the FDA had a blind experiment that had to be run where there was a training set that had to be used for calibrating a model, and then some held out data that nobody had ever seen. The FDA sequenced using the low throughput, very expensive method I was talking about before. So we know, with a very high degree of confidence whether there's a variant present in the sample or not. And then that ground truth data was then compared to high throughput sequencing outputs that were processed with some sophisticated computational algorithms, right, to try to identify the variants. And DeepVariant won this competition on identification of locations of variation within the genome. And we got second highest performance on the places where there were some deletions or insertions of additional sequences. You can read about this more on the Precision FDA website if you care. And late last year, we published the source code for this thing. So it's already like a very popular project, especially in the area of bioinformatics. People are downloading and using it. Again, it's open source. It's built on TensorFlow, which is open source, so you could run it on your own hardware. There's no dependency on Google to do any of this. You can also run it on Google Cloud, though, as a service. We have a managed service that uses accelerators like GPUs or tensor processing units under the hood to make it run fast and cost effective. And that's the type of work that I am doing these days, is applying this to other problems that are of interest. So, for example, I'm doing some work in agriculture these days. This figure I'm showing you here is a picture of some corn on the far right. That's the modern corn. Over the last 10,000 years, that was developed from the thing on the far left. This is a time series, basically. So there was some selective breeding that was done where, humans saw this thing, were able to eat some seeds from the thing on the far left, and liked it. And they might have observed that if they grew it, some of the individuals were a little bit bigger, or had more kernels, or fatter kernels, or were more nutritious or drought tolerant or pest resistant or something, and selected for those. And so over time, we're able to develop it into this thing on the far right that's a staple crop of the world today, and it feeds a lot of people. We need these improvements in this crop or other staple crops around the world, like rice and sorghum and potatoes, and there's a number of starches. This is where we get most of our calories. We need it to basically match the general economy's growth rate. So it's growing at 3% per year. That's exponential, right? It's not a big exponent. It's not like squared or something, but it is exponential. And you can imagine that if you want to keep getting those level of yields, it gets exponentially harder, and you have to throw more and more resources at it to get a little bit more yield. So getting back to emojis and back to some of the genetics stuff that's the context of this computational genomic analysis problem we were just talking about using deep learning machines, you basically want to make sure that you're crossing the right parents to produce the next generation of offspring, and you want to make sure that offspring that you've produced, you give it the right environment to maximize the yield for whatever you're aiming for, whether that's like, number of grams of chicken meat you get per unit time, or whether it's resistance to avian flu outbreaks, or this is the case animal examples I'm giving you now, but it's basically the same problem. You could even imagine that humans are doing this kind of things to themselves. But we're basically interested in identifying these functions underlying this process here, right? Yeah, and this is like real industrial stuff. So this is actually out of an SEC filing from Monsanto. I forget what year it was. I didn't add that. But there's basically this cycle that happens where there's some sampling, some DNA extraction. You generate a genetic marker fingerprint from a given sample, and then figure out what individual you want to combine that one with to make a new progeny for the next generation, moving through time to improve the quality of the crop, right? So this is called, generally that's called selective breeding. And then once you start using high-resolution genetic markers, as opposed to just looking like at the size of going back here, looking at the phenotype and saying, oh, this one is like a bigger individual, so I could cross the two big ones to make an even bigger one, that stops working after a while because any given individual has environmental components. So there's some like bell curve distribution where it might genetically not be so big, but just happens to have become large through environmental factors, right? Noise, effectively. But if we start doing this kind of process where we incorporate the information of the genetic markers into the process, that becomes very high-resolution information and can be highly predictive because it's the genetic basis for the phenotype that you're observing. So this is what's happening today, and this is how we're keeping up with this exponential growth that the economy demands to make sure that we keep making enough food, right? All right. Yeah, so that's in general what's going on. This is the first application of using deep learning technology to categorize genetic variants in a given population. So this is, I'm working with the International Rice Research Institute. They're based in Los Baños, Philippines. And it's an international organization that works with all of the departments of agriculture around the world that for countries that grow rice. And so that's, I forget how many, but it's all over the place. Those departments of agriculture have their own local varieties of rice, and they're doing their local optimization. Periodically, they send seeds to IRI, the Rice Research Institute, that then does some centralized optimization, and then ships seeds back out to the participating agriculture departments so that they can benefit from it's kind of like a global scatter-gather approach, right? So everybody's optimizing locally, and then there's a single global optimizing node. So the central node has a lot of data, as you might imagine. They have a biobank of more than 100,000 different types of rice. They also keep these in that under the glacier in Norway, in the biobank in Svalbard, right? They have some copies there. But what we're interested in doing is looking at a subset of these samples that are well characterized. There's about 3,000 of them. They're highly inbred. So that means you can take rice and you can cross it with itself. So you can have a male and a female cells from a single rice plant and cross it, and then all the offspring basically have only one genetic parent. And so if you do that repeatedly, what will happen over time is that all the genetic variation will be washed out and it becomes identical to itself at all positions. It's called homozygous, right? If you have some individuals like that, they are genetically there's not very much, they're genetically very predictable because there's no genetic variation within that given individual or given set of individuals who are from the same parent because you've inbred them many times. So if they'd been inbred many times, you would expect that the heterozygous rate, or the number of positions in the genome where the bases are different from one another on the chromosome, should be very small because you've inbred many times. What I'm showing on this plot here is a previous method on the x-axis versus the DeepVariant deep learning-based method on the y-axis. And if they were performing equally well, you would just see basically some kind of smear along the diagonal, meaning that they basically have approximately the same amount of noise in their outputs, or are modeling approximately the same, like it's not biased in any way. But you can see there's actually some bias here, where DeepVariant is actually not calling very many heterozygotes. It's mostly homozygous, which is why that point cloud is mostly horizontal here, because the previous best method, which is labeled GOTK here, is actually making a lot of calls where it thinks there's some genetic variation present or a false positive relative to what we know because the plant is inbred, relative to DeepVariant, which is not calling so many of these false positives. And so we look at the ratio of this, we can basically say that there's about 6 to 10x over-representation of variance within these 3,000 individuals we sequenced relative to what we know ought to be there. We know there ought not be any variation observed because of the inbreeding, but we observed a whole bunch of genetic variation, gotta be false positives, okay? That's important because if we know that we need to have good markers for a given individual to keep improving at 3% per year, right, exponential, if you get garbage going into that system, garbage in, garbage out. This is basically giving false information to the breeders so that they can't actually select the right individuals for breeding to produce the next generation of plants. So we're giving more leverage to the breeders by doing this, by giving them higher fidelity information about genetic variants, improve food security. All right. So that model I was showing you, this one, this result for rice, was actually trained on human data. Because remember, we have very high quality data from humans that we don't have from other species because humans are generally willing to spend money on themselves and not willing to spend money on plants and animals. Just sort of like how it is, right? We're more important than anything else to ourselves. So there's not a lot of funding that goes to these other species, but we're demonstrating here basically that we can train on the high quality data and port it over to other species. So you've got mice and rats being used for disease modeling for building drugs. Well, we can carry over this method and apply it to those species and don't have to produce high fidelity data for mice and rats. It even carries over to rice now, so that's very far away. It's not even an animal. And we're finding that it performs better than the current best practice. Assembly agnostic, I don't want to talk about that. It's basically just some other technical stuff. The reason that the technology is getting so cheap is because there's new different types of chemical parallelization techniques that are being developed. And every time we build a new one of these things, it's a completely different data type with different types of biases that need to be tuned by PhDs with 20 years of experience to make sure we can eke out a little bit of performance when we're trying to wash out the noise and identify the true genetic variants. It turns out that we can also use a model that was trained on a previous generation of the technology. It can be carried forward. So we don't need to collect a bunch of data from the next generation of technology to keep using this deep learning model. It's basically very general purpose, which means that when the next generation of tech comes out, sequencing tech, we can use the existing deep learning model, which is learned from a bunch of previous generations and can be extrapolated out to the next generation. So it saves a bunch of money and time there as well. The next thing that we're doing, I actually went to the Philippines. This is a photo from one of the rice fields. We want to start combining the genomic, like variant information with environmental information so that we can basically digitally model what's going to happen if we have a known set of genetic inputs and a known set of environmental inputs. What is the expected output? Because now we have all the variables in our hand, right? So here's an example. Each one of these little white stakes you can see here is demarcating one by one. It's a 1 square meter of space. And inside of that 1 square meter of space is, I forget how many plants, but it's the same number of rice plants in every square, okay? And every one of these squares actually contains the same type of rice plant. They're genetically identical. It's the 3,000 I was showing you earlier. So it's a different inbred individual in each cell, pixel basically. They're imaged by drones. You'll see where I'm going here. On the right side is a wet condition for rice, and on the left side is a dry condition. You can see it's basically kind of flooded on one side and very dry soil on the other. It's the same individuals in both of them, where we've given them 2 different environmental conditions. And then what we do is over time, we take a drone, put it up in the air, and take high-resolution pictures of the field over time. So then what we know is basically, how did the rice grow? What were their properties? Did they fall over when it was windy? It's basically the same wind across both fields, right? They're right next to each other. They're getting the same amount of sun, same amount of rain, etc., but they're going to have different phenotypes, obviously. You can't really see it very well here, but there's a dramatic amount of variety between each of these cells in the field. And so we're starting to collect basically time series of phenotype data where we have environmental sensors for, again, all of the weather data, right? Plus we know all the genetic data. So we can now combine all of that to further make sure we know what, not only what individuals we should be crossing to make the next generation, but also knowing if we can forecast the weather and we know next year is going to be extra hot or extra dry, maybe we should be planting a particular type of seeds to make sure that we produce the right amount of rice, right? So this is basically managing yield, reducing risk, starts merging into the insurance industry and so on at that point. Yeah. Oh. Yeah, sure. I'm basically done. I'm doing this on cannabis right now as well. We've got a bunch of like several thousand cannabis species coming in. Those are really interesting because they grow typically in an indoor environment, right? Highly controlled with sensors and IoT devices, easy to put into the cloud, and then mosquito sequencing also. So those are interesting because we want to stop disease spreading. All right, so that's all I've got. Thanks.